Hi, I'm Laura Shannon. I'm an assistant professor of potato genetics, genomics, and breeding in the Department of Horticultural Science here at the University of Minnesota. Today, I'm going to be talking about a few approaches my lab takes to autotetraploid breeding. Specifically, I'm going to talk about sequencing, math, and avoidance. So first, I want to introduce you to russet burbank. This is the most commonly grown potato in the US, and it is over 100 years old. This is in contrast to other crops where new varieties are released and regularly replace older varieties. In general, in potato, the varieties most commonly grown are 50, 60, 70, 100 years old, and we've seen basically no increase in yield in potato in the last 100 years. This is a problem because potato is really important. It is the most commonly grown vegetable crop in the world. It is the third most common food crop eaten in the world. It is central to the diets, cultures, and economies of countries all over the world, and of Minnesota in particular. So we'd really like to be able to regularly develop new varieties that allow us to respond to changes in the climate, changes in the environment, and changes in consumer demand. So why are we so much slower with potatoes? Well, it's because potatoes are difficult. So there's three main things about potatoes that make them hard to breed. The first is they're a horticultural crop. So there's just a lot of breeding targets. We're not just interested in yield and disease resistance. We're also interested in skin color and skin set and size and shape and depth of eyes and whether or not the skin stays attached and how long you can keep them in storage before the fries turn black when you fry them. People have developed varieties which yield way better than russet burbank, but they don't meet these other criteria that growers and processors have, so they haven't been widely adopted. So these multiple targets is one challenge. Another challenge is that potatoes are clonal, and this is a problem because it's a problem for disease control. Sexual reproduction clears out diseases and clonal reproduction doesn't. It's also a problem for ramping up seed for new varieties, especially early in their life cycle. And finally, potatoes are autotetraploid, and that's really what I want to focus on today. So if it's been a while since you've thought about autotetraploidy, I just want to remind you what it is. It is having 4 copies of each chromosome, all of which can pair with each other and indiscriminately. There's no preferential pair. So if you have 4 copies of each chromosome, then you have 5 possible genotypes at a biallelic locus. There are 2 homozygous classes, just like for a diploid. You can be homozygous pink or you can be homozygous yellow, but there are 3 different heterozygous classes. And this makes things complicated. It makes things complicated for genetics, and it makes things complicated for breeding. Specifically, it pulls some really important breeding strategies off the table. So the biggest issue here is inbreeding. So if you inbreed a diploid for about 7 generations, it's pretty much homozygous. To get that level of homozygosity in a tetraploid, you have to inbreed it for more than 28 generations. That's basically impossible on the timescales we work on. So we really just don't. This is not only a problem because we can't create inbred lines that take advantage of heterosis. This is also a problem because it means that we can't change existing varieties. We can't use introgression. So we couldn't introduce a new disease resistance trait to russet burbank. Russet burbank just is what it is, and every time we cross it, everything gets shuffled, and it's all totally new. What this means in the field is that when we do crosses, full sibs look wildly different from each other. So these are a bunch of flowers from full sibs. And you can see here that they have all different colors, they have all different shapes, there are different flowering times. Some of them just don't even flower. So our year one in the breeding program is we do crosses and we plant them out, and we plant out 20 to 60,000 distinct genetic individuals, and we go through and we do visual selection. And we select about 1% of those because most of them don't look like potatoes or don't fit into a market class. So you can cross two russets for fry processing together, and 99% of what you will get out does not look like a russet because we are just shuffling the whole genome every time. And so that makes breeding slow. Another thing that makes breeding slow was highlighted recently by Patrick Monahan and Genevieve in a recent paper. And what they're showing us here is that the response to selection in tetraploids in orange or in octoploids in purple is just so much slower than it is in diploids, which we see here in green. And this is important because selection is what we're doing in breeding. And so it being just inherently slower slows us down. So the really exciting thing is that after 100 years of russet burbank, right now we have a bunch of new technologies and strategies and approaches to breeding potato. And these are throughout the potato community of breeders and geneticists. And I'm going to talk about three of the big ones that our lab is involved with. I'm going to talk about sequencing, talking about some of the genomics approaches. I'm going to talk about using mathematical models to do genomic selection. And then I'm going to talk about just avoiding tetraploid breeding altogether by creating diploids. Reinventing a crop is a community-wide effort. Breeding is always a community-wide effort. And so all of these things are things we're working on with a lot of collaborators who I will talk about as we go through. So I want to start by talking about genomics. And as I mentioned in the beginning, autotetraploidy complicates genomics. And this is an image from Illumina explaining their array. And so what this shows is how you identify genotypes from binding on an array. So in red, we have one homozygous class, in blue, we have the other homozygous class, and in purple, we have what would be the heterozygous class for diploid, and then everything else would be error, right? But if you have multiple heterozygous classes, then you have to distinguish between them all. And you get smaller boundaries between them, and it's harder to distinguish what is a distinct class. And then to assign an individual to a heterozygous class. I used this picture of the genotyping array because I think it's a really clear illustration, but we have the same problem with sequencing. The more heterozygous classes you have, the harder it is to distinguish between them. And so I want to talk about some data we've been looking at that I think really illustrates how difficult sequencing can be in autotetraploids. So this is a project that was started by Cindy Tong and Corey Hirsch, and they were interested in finding genes for skin color in potato. So there was a clone, Norland, that was commonly grown for a while. It's a red potato. And then someone in the field noticed that there was a darker one that was redder. And then that became a new clone called Red Norland, and that's the most commonly grown clone in Minnesota. There's also a sport of Red Norland that's even darker called Dark Red Norland. So all of these are basically clones of one another. There presumably were a small number of somatic mutations between them that create these different clones. So Corey and Cindy's plan was they would sequence all of these. They sequenced them to about 21x, which is pretty good. That's basically de novo sequencing for a dog. They sequenced them to 21x, and they thought they would identify the differences in sequences, and the differences in sequences that were in the anthocyanin pathway would explain the differences in color in these clones. So when they looked at the data, there were hundreds of thousands of differences between each clone and the other clone. And that is when our lab got involved, and the analysis on this in our lab has been done by Muhyiddin Youssef, who's a grad student in the lab. And so we saw all of these differences, and we were trying to figure out why, because these are clones, right? There shouldn't be this many differences. There should be a small number of somatic mutations, but 183,000 sounds like a pretty large number of somatic mutations. So our first thought was maybe it's a coverage problem. And so this figure is from Sushan Rue in Jeff Endelman's lab at the University of Wisconsin. And what they were doing was they were looking at how much coverage you would need for GBS to be able to confidently call genotypes. So in Coral, at the top, we have a diploid heterozygote. And using 5 to 15 read depth, you're at like over 95% accuracy. And once you get past 15 read depth, you're basically at 100% accuracy. You can consistently call those diploids. However, it's more complicated for tetraploids. You need more read depth, and it really depends on the genotype you're trying to call. So the easiest genotype to call is a nulliplex, which is in turquoise, and that is like 4 A's. That's a completely homozygous genotype. And then from there, you need more and more coverage to call a tetraploid simplex genotype. So like AAAB. And the most difficult thing to call is a tetraploid duplex genotype, which is AABB. And so what happens is that at low coverage, these heterozygous classes can be miscalled. And so that seemed like the most probable explanation for what we're seeing, is that at low coverage, because we're only in 20x coverage range, in which we expect like 60 to 70% accuracy on SNP calls. So some of these SNPs are just being called wrong, and there looks like a SNP when there isn't. So to test that, we compared 2 different kinds of SNPs. So when the differences in genotype were between 2 heterozygous genotypes or between a heterozygous and a homozygous genotype. And we looked at how those were distributed over read depth. And so you can see here that in green, where we're looking at the heterozygous genotypes, they are really clustered toward the low end of the read depth spectrum. And the homozygous heterozygous transitions are in sort of more the middle of the read depth spectra. So this is consistent with what we'd expect if the problem really was this coverage problem. We also compared our Norland to published Norland data. So we have 12x Norland sequence from Hartigan et al. 2017, which was in PNAS, and looking at their Norland and our Norland, which again should be exact clones of each other, we saw over 1 million differences. So there's a couple of possibilities here. One of them is that clones generate millions of somatic mutations. But that seems pretty unlikely. One of them is that something really weird happened in tissue culture. That is possible, but also pretty unlikely. The most likely explanation, seems to me, is that we are just miscalling heterozygous SNPs, and that's introducing differences between clones we wouldn't see otherwise. So this is a problem. Not being able to sequence tetraploids has some real limitations, and it means that we haven't done even some very basic gene discovery work. For instance, the most commonly grown market class in the US is russets for French fry processing, and we don't know what gene causes russeting. So being able to sequence potatoes would really open doors for us in terms of identifying the really important genes in potatoes. So we've been thinking about how to solve this as a community, and what we decided on was using the Energene pangenome technology. And this is a picture of how that's supposed to work. It's from a recent paper out of Candy Hirsch's lab. And so what you can see is that you sequence a bunch of different individuals, and then instead of comparing any one individual to a reference genome, you can compare newly sequenced individuals to this graph pangenome, which takes into account the known genetic differences, the known genetic diversity, right? And so the idea would be that then we could sequence things at like 20x coverage, and we could align them to known haplotypes. And first of all, it would help us phase. And second of all, it would help us correctly call SNPs. So that was the plan. And we put together an international collaboration. We have a huge consortium that's all working on this. It's expensive, it takes a lot of work. And the person running this consortium is Richard Finkers out of Wageningen. But a lot of the work has been done by a grad student, Genevieve Hoops, in Robin Buell's lab at Michigan State. And so we all agreed that we would sequence 6 clones. 4 of them are Dutch, and they're split between starch clones for processing and fresh market clones. And then in the US, we contributed 2 clones, Atlantic and Castle Russet. Atlantic is a chipping clone, so a round, white-fleshed, white skin, real thin skin clone. And Castle Russet is a russet, so that's like a big potato that we use processing for French fries. What Energene produced were tens of thousands of scaffolds, and that is the state that the 4 Dutch clones are still in. But we have 2 phased pseudomolecule assemblies that Genevieve and Robin created for the 2 US clones, Atlantic and Castle Russet. And we were able to do that because we had phased genetic markers for them using GBS data, and that was a combined project of Jeff Endelman and Dave Douches and Sagar Sithavali and our lab. And then Robin and Genevieve contributed Hi-C and Oxford nanopore data. And so we were able to get these pseudomolecule assemblies for 2 clones, which is more like the sort of graph genome we were talking about originally. So there were a lot of steps to this that I don't have time to talk about, and there are a lot of findings coming out of here, but I really wanted to highlight the one that I think is one of the most interesting, and this is Genevieve's work. So these are the Busco findings. So this is, they looked for a set of commonly occurring genes in the genome, and they checked if they were present and how many copies they were present in. And the vast majority were present in duplicate copies, which is exactly what we expect for a polyploid. And then we can further break that down into how many duplicate copies. And we can see that some are in single copy and some are in double copy, but most of them, the majority of them, are in 3 copies for both Atlantic and Castle Russet. And this is true of the 4 European clones when you look at them too. Almost everything is in 3 copies, which means there are 3 different alleles per locus for all of for the majority of the most common genes in the genome. And so that's sort of what we expect in general, that there are 3 haplotypes per locus for potato. We cannot distinguish with this analysis. Genevieve has done further analysis that allows her to distinguish, but we can't distinguish between if the 4th copy is missing or just identical to one of the 3 copies. So ABC missing looks the same as ABCC here. But really what this is telling us is that potatoes are just incredibly, incredibly heterozygous. So having a pangenome lets us look more in depth at particular loci. It lets us look at genomic patterns across the genome, and it also lets us learn about the history and diversity in potato. And I just want to talk about one case study for that, which is from Dr. Xiaoxi Meng, who's a postdoc in my lab. And we were really interested in looking at introgression. So potato was domesticated in Peru at least once, and it's not really clear what the ancestral species is. It could be a species complex. But there are just a ton of wild potatoes around there. And we sort of think there might be a history of introgression, and we know there's a recent history of introgression where breeders have used wild potatoes to introduce disease resistance, especially. And so what Xiaoxi did was take these genomes and look at the genetic distance between them, so tuberosum, and which is cultivated potato. So look at the difference between them and cultivated potato and the series of wild species on the right-hand side. And as you can see, there is just a ton of introgression. And so the introgression in this case is regions of the genome that are more closely related to wild species than they are to other cultivated species. And you can see that they're spread across the whole genome. So then we wanted to see if they had any of those regions in common, if any of them were consistent. And we can see that there's, about 50 here that are found in all 6 accessions, all 6 cultivars we looked at. And they're also found on every chromosome and spread across the genome. So this suggests that there is a history of introgression too, right? And so if these were old rather than just coincidence, we would expect them to be on small introgressions. So the longer an introgression has been in a genome, the smaller it is. And these shared introgressions are smaller than the introgressions in general, which suggests that these might be introgressions from before potatoes moved into Europe, and suggests this long history of introgression. We also know there are more recent introgressions, and one of the biggest studies in introgression in potatoes so far has been using the SNP array and dating the SNPs from the SNP array. So Herman van Eck's group put together a list of SNPs from the SNP array that don't appear in any varieties created before 1945. They only occur in post-1945 varieties, and that is when people started doing introgression breeding in Europe with wild species. So the idea is that those are introgression SNPs. And so we looked and saw if those were present in our 6 genomes, and we found they were. Those are the gray dots. And then we looked at the overlap of those with our introgression regions. And the green dots are introgression regions that were at least 5 kb, and the yellow dots are introgression regions that were at least 10 kb. And so we do see a lot of overlap in basically everything but spunto, where there aren't that many introgressions to begin with. And as we said, the shared introgressions are shorter. The introgressions from these post-1945 SNPs in them are all longer than other introgressions, are on average longer than other introgressions we expect. So this is consistent with that there are these multiple timescales for introgressions, some which are much older than others. And so this, I think, is really exciting because it tells us where some of the diversity and heterozygosity in potato is from. And it suggests that there has been this really long history of introgression in potato, and this relationship between cultivated potato and the wild species that's been around. And this is just one example of the kinds of things we can do with a pangenome, and why having sequence and being able to sequence autotetraploids is really powerful. So next, I promised I'd talk about math and building mathematical models. So we're going to talk about genomic selection. And I don't think I need to tell this audience, but genomic selection is a way to use mathematical models to speed up the breeding process by predicting which combinations will be good parents, what individuals will make good parents. So the breeding process for potato is long. It generally takes 7 to 12 years or more from crossing to release. And in general, breeders tend to decide to use something as a parent in the 7 to 12 year range, which is slow, and it slows how fast we can make genetic gain, especially when you combine it with all the other things going on in tetraploids. So what would be great is if we could use genomic selection models in genomic prediction models to start picking parents earlier in the cycle. This is a well-known, well-established technique in a lot of crops. It is a recently established technique in potato because, once again, autotetraploidy makes life hard. So here's sort of 2 examples of the ways in which it makes life hard. So on the left, and both of these are out of Jeff Endelman's lab. So on the left, we are looking at the different kind of dominance you can have if you have 3 heterozygous classes. So there's basically one kind of dominance in a diploid, and there are multiple kinds of dominance in polyploids. And then on the right, we're looking at the variance explained by different additive and dominance models. And so understanding these different kinds of dominance and being able to track them in your population, and also getting a really accurate kinship matrix, which takes into account the relationship between these different heterozygous classes, is really important for accurately predicting phenotypes and genetic effects in these kind of models. So the first thing we had to get right was genotyping. And we have a couple of different systems for genotyping now. So the first is that Dave Douch's group at Michigan State has created a 31K array on Illumina. It's great, but it's really expensive. So it's $50 a sample, which is kind of too much for genomic selection. Another option is GBS. It works in tetraploids. We have calling models, but because of how high of read depth you have to get, this is also kind of expensive. So the sort of solution to this we have is that the US breeders, as led by Jeff Endelman, have paired with the Excellence in Breeding program out of SIP to use a dark tag array, which gets us more like 1,000 to 5,000 SNPs for much cheaper. And so that is really going to help us move forward. Sort of a similar technology to GBS, but it uses set markers. So we are able to use it more like an array. So you need genotyping data to build a model, and you need phenotyping data to build a model. And this is a picture of me phenotyping red potatoes for color and how well they keep their skin and shape. And I am just writing 1 to 5 scores on a clipboard, which is less than perfect. And especially because, like we talked about in the beginning with Genevieve and Patrick's findings, because selection is slower in autotetraploids, it becomes even more important to get really precise phenotypes we can put as much oomph behind selection as possible on. So we are interested in developing more accurate ways to get phenotypes. One of the tools we're interested in using is image analysis. And so we have been taking a lot of pictures of potatoes in a light box, following Dr. Maria Carrazza-Harder's method from Jeff Endelman's lab. And this work in my lab was started by Dr. Carrie Schmitz-Carly, who has since moved on to breed diploid potatoes, and is being continued by a new graduate student, Michael Miller. So the first thing that we have done with image analysis is to identify color and skinning. And so one of the things we can do is we can take from an image is we can take a median color value for every tuber. And then also we can identify regions where the tubers have lost skin. So those are shown here in yellow. And we can quantify them way better than me with a clipboard saying, like 20% is gone. Showing we can quantify these way better, we can improve heritability. For skinning, we have improved heritability from 0.62 using a visual rating of skinning to 0.68 using our image analysis, which we're calling TUBAR. Color, we get a 0.58 heritability when we're just using a visual rating, but we can break down color into 2 components using our image analysis, redness and lightness, and our heritability for redness is 0.63 and our heritability for lightness is 0.62. So these are really important gains if you're considering that selection is going to move slowly anyway. So we're interested in expanding this to other phenotypes. One we've been working on is tuber shape. So this is the chart I was using to analyze to assess tuber shape. And so now what we're doing instead is we are taking the perimeter and area of each tuber. We're taking the maximum length, and we're creating a minimum bounding box, which we can get a length and width for. Then from there, we can measure compactness, which compares the area and the perimeter. And we can measure roundness, which compares what we have to a convex hull. And again, we're able to really improve heritability. So compactness is a little less impressive than breeder shape, which is just me evaluating it. But we are really able to improve heritability in terms of length to width ratio, and roundness. So again, this really gives us more power to select for these traits. We're interested in expanding this further. We're working with Darren Hagenson at the USDA in East Grand Forks on this. And again, this is Michael Miller's thesis project. So we're interested in looking at chip and fry color which is important for marketability. We're interested in looking at pressure brews, and we're interested in looking at blemishes due to disease in the field, like scab and silver scurf. If you are interested in sort of this aspect of what we're doing in quantitative genetics of autopolyploids and using that in breeding, I highly recommend 2 talks we have coming up in the horticultural science seminars. On April 14th, Bodhi Alokalu is going to talk, and he's done just a lot of work on genotyping technologies and mapping technologies for autopolyploids. And then on the 28th, Oscar Riera Lizarazu is going to talk, and he's the leader of an SCRI we have for building polyploid tools and creating multi-environment prediction models for potato. So then the last thing I said I'd talk about is we can address tetraploid breeding by avoiding it entirely and turning potatoes into diploids. And this is just a huge international effort. People are going at it in a lot of different directions. And I'm going to talk about how we're doing it in the US. And this is a picture of the SCRI planning meeting for diploid potatoes. So there's a lot of people involved. So the vision here is that we will be able to convert potato into a diploid inbred crop, inbred hybrid crop. So that we can take advantage of integration and heterosis and all of the powerful tools of diploid breeding. So how we are doing this is we are going out to the field and we're collecting vans full of flowers, and we're bringing them back to the lab, and we're pollinating them with a diploid inducer, which at low frequency jettisons its own chromosomes and creates a seed which just has the gamete from the tetraploid maternal parent. So what we wind up with are these plants that are plants, but diploid with half of the chromosomes from a tetraploid. And so we do all these crosses, we plant that, we keep the flowers in big tubs, we collect the fruit, we identify diploids, and this is a really low efficiency process, but it does result in diploid potatoes. So from there, then the diploids are dihaploids, are haploid diploid potatoes, are crossed to a self-compatibility donor, which allows us to start inbreeding. And our lab has only been doing this for 3 years, so we don't have very impressive inbreds yet, but these are the inbreds from our collaborator Jeff Endelman's lab in Wisconsin. And you can see that some of these are pretty impressive potatoes. Some of them look kind of crappy, but we do have ones with nice size and shape. So this is a slow process, but it is resulting in some promising potatoes. So I just want to talk specifically about one set of phenotypes to talk about why we think this is going to work and how we think this is going to work. And so this is from a collaboration with Dave Eichholt from PepsiCo, and this data was gathered by Hussein Aga, who is a graduate student in the lab. And so we were interested in distinguishing between diploids and tetraploids. We were hoping to use it to identify tetraploids from the original crosses that we could throw out and just keep the dihaploids. And our thought was that they would have different absorbance spectra using NIR because they have different amounts of DNA, and we assumed that would involve different amounts of chemical compounds in the leaves in general. And so we looked at these, and what we saw is that the averages are different, but that they don't separate or differentiate anywhere on the spectrum. There is overlap everywhere on this whole spectra, and there is no way to make a clear division. Hussein's been trying all sorts of machine learning models, nothing. So then we wanted to see, well, are they just the same? Are the means really different? And so this is a Manhattan plot of basically t-tests at different bands along the whole spectrum. And we have two cutoffs from a permutation test. And so what you can see is that in many places along the whole spectrum, we really do see these significant differences. The means are significantly different, but there's still this overlap. And so this is an example of this, looking at 2200.3, which is one point on the spectrum where they look pretty different. And we can see that the means are different, it's significant, but there are these overlapping diploid and tetraploid distributions. And in fact, there are a small number of diploids on the extreme end of the tetraploid distribution. So what this is telling us is that even if diploids and tetraploids have separate phenotypes, even if diploids look way worse than tetraploids on the whole, there are still a few diploid individuals that can recreate tetraploid phenotypes, which I think is really promising evidence that this diploid breeding can work. We just have to do it at huge scale. And so that's what we're doing. Like I said, people from all over the world are working on this. They're working on this in China. There are a ton of European companies working on this. The US breeders have all banded together to work on this. But it's really a project in its infancy. So the most optimistic estimate I've heard for when we might start to see diploid potatoes on the market is Ethiopia in 6 years and Europe in 10 or 12 years. So we're probably talking 20 or so years until diploid potatoes really become the new potato, right? But they have a lot of potential, and what they'll allow us to do is respond really quickly to and create new varieties quickly. So to changing climates, to changing environments, to changing consumer pressure. And so this seems really worth doing, even though it's going to take a long time and a lot of potatoes. So in conclusion, we've been growing the same potato for the last 100+ years in the US. We've been growing russet burbank. But now is a really exciting time to be a potato breeder, because we are developing a whole lot of new technologies and new strategies to really allow us to change that. And so I talked about 3 of the ones we're using in our lab. I talked about using sequencing and developing a pangenome to allow us to better understand the intricacies and the history of the potato genome, to better understand important genes in potato, and to use that and to be able to leverage that knowledge for breeding. I talked about math. I talked about building mathematical models to allow us to shorten the breeding cycle and pick parents faster using genomic selection. And then I talked about speeding the breeding process by dispensing with tetraploids altogether and just creating diploids, which is a slow approach now, but will be a fast approach once we get it working. And so I think that these are all strategies that are really going to allow us to start eating new varieties of potatoes. Breeding is a huge collaborative effort. And I just really want to thank my lab. I want to thank our collaborators, some of whom I named through this, but honestly, we get a lot more help than I could talk about in 45 minutes. And then I also wanted to thank our funding agencies. And thank you very much for watching this. And I really look forward to talking with you all on Friday. Bye.